So yeah, greetings from Massachusetts. I have my virtual background on. This is our beautiful campus behind me in Worcester, Massachusetts. We're about an hour west of Boston. And today I'm just gonna kind of highlight and talk about some work over the past number of years and what we're kind of doing more recently with regards to microswimmers and in particular with applications to sperm motility. Okay, so yeah, as I mentioned, this is kind of I'll be talking about a few different projects here and there. A lot of it started with Lisa Fauci and Ricardo Cortez at Tulane a number of years ago, and I'll try and give credits to appropriate people as we go along. Okay, so in general, I've been very interested in thinking about kind of moving elastic structures, such as microswimmers at lower zero Reynolds number. And in particular, really thinking about how this moving tail, this elastic tail couples to, or the emergent kind of beat forms of these tails for different conditions. So this is a very classic picture. It's seen quite often in a lot of papers by Ho and Suarez. And it's, bull sperm, a snapshot of time away from each other, kind of in something like water. On the left side, then we move to something where they change a chemical concentration, calcium inside the sperm flagellum, and it starts kind of beating hyperactively. With this large asymmetric bending, and then you switch again to a more like a gel or a viscoelastic fluid or environment, and the dampening of these asymmetric beat forms happen. So we've thought a lot about, or I've thought a lot about kind of these different behaviors. So here is this kind of hyperactivated or this high calcium case in something like water. And here is, a actual calcium fluorescence along the length of the flagellum to show how it's changing, in time and space. And lower calciums are the blues, and that corresponds more with kind of symmetrical beating, where the reds and yellows are highly asymmetrical beating. So, a lot of what we've been thinking about is, how do you develop good models to couple the kind of calcium dynamics or kind of the relevant kind of biochemistry. How do you get the right modeling frameworks for, the beating of these tails? How do you, develop good numerical methods to do this? And, kind of the application side of things, how does the sperm reach the egg? Can we think about artificial microswimmers to, get to certain locations and swim in a certain way in different fluid environments? And we'll get back towards that at the end. But as I mentioned, right, so there's a lot, and I know a lot of you guys on this Zoom session right now are very interested in different aspects of swimmers or low Reynolds number, and kind of the different aspects that we've also been looking at. But the way I see it, there's a lot of current challenges. There's a ton of great being work done, but also a lot of open questions that still remain. What are the right ways to do these multi-scale models? How do you really get kind of fully 3D geometries and couple everything in the right way? And really thinking about kind of these efficient numerical algorithms. And I'll touch on, a few things as I go through some kind of highlight some different projects over the past years. In terms of how I view these microswimmers, or in general kind of sperm, I think about this elastic flagellum. The sperm flagellum itself has this very complicated structure with kind of 9 microtubule doublets. There's kind of these dyneins, or active force generators that are walking along the different microtubules, which kind of causes bending relative to one another. For these different kind of species of sperm, depending on whether it's mammalian or invertebrate, there's different additional structures. And also depending on the species, some of these kind of microtubule doublets have fused together, and then you can get more of a planar or non-planar, quasi-planar, or helical beat form. So the way we thought about things is really kind of trying to simplify a lot of this complicated structure of the sperm flagellum into a very simplified representation. So really going from, these complicated interior dynamics to kind of a really collapsing everything to a center line and doing as best we can with just a really simple center line representation for kind of computational ease. And then we think about this actuated elastic structure, whether it's a rod or a center line or a filament. We think about immersing that in a viscous compressible fluid. So we have kind of an immerse boundary type approach. And we think about the fluid feeling the swimmer through this forcing term. And we think of our structure, let's call it big X, it's moving at the local fluid velocity. Okay, so in terms of kind of solving these different equations, or our fluid flow equations, right? So I have incompressible Stokes equations here. To solve those, and with these kind of moving structures, there are a number of different options. Not limited to slender body theory, resistive force theory, immerse boundary method, boundary method, and the method you choose, of course, will depend a lot on what you're trying to do. In general, we've been using a method of regularized Stokes slits developed by Ricardo Cortez for a variety of reasons. But I like the method of regularized Stokes slits because we have these fundamental solutions. So for some point forces, or if I think about my really simplified structure here, if I kind of discretize my swimmer or my filament into some point forces, I can think about creating a Lagrangian method. My point forces, I can think about regularizing them, and I can get a Lagrangian method to have fundamental solutions, and through this kind of superposition of fundamental solutions, have a solution for the fluid flow everywhere on the structure and off that's everywhere incompressible. And the beauty of this is there's lots of, so this is a very kind of simplified description of the method, but what's great about it is there's lots of flexibility in terms of the numerical methods you want to use, how you want to discretize your structure. There's flexibility in the force models that you want to use. There's also flexibility in terms of whether you want to use an infinite fluid domain, which are going to be kind of most of the results that I'm showing you today, or whether you want to think about periodic domains or domains with walls. We can easily handle them for the most part in this kind of method of regularized Stokes slits. Okay, so in terms of the swimmers that I've been thinking about, we have these filaments, and as I said, we're trying to collapse a lot of kind of biological and mechanical information onto these slender structures. And from a computational perspective, just having a center line or this filament is very nice, instead of modeling the entire kind of cylindrical flagellum or modeling all of those microtubules and dyneins. So what we do is think about having this, this filament or this center line. It's bending, or it's going to be actuated. So we want to think of these swimmers as potentially propagating some type of beat form on average, and we think about deriving some type of force model that's going to be based on an energy argument. And the energy argument says that on average, we want to kind of maintain an extensibility, and the swimmer is going to want to bend a certain way, and we decide on that model. But we devise it based on kind of a tensile and a bending energy. And in the absence of other swimmers or walls or other things, on average, the swimmer is going to try and minimize this energy and try and maintain the bending that we're prescribing. When we do this, we have a set of parameters, so kind of a tensile and a bending stiffness. And with a lot of these models, you can think of these parameters in a few different ways. They can have a very numerical or kind of analytical type of thing as more of a interpretation as more of a Lagrange multiplier. So how large you choose this will depend on exactly how strictly this equation is satisfied. On the flip side, we can also think of it as kind of a modeling parameter and back out for a particular tensile or bending stiffness, what's the flexural rigidity or kind of energy in that system. So we do that. So we think about this kind of modeling framework. We know for these different swimmers, there's a very well-known kind of stiffness. Some of it varies along the flagellum, depending on the species. We've done constant stiffnesses, variable stiffnesses. But if I just kind of use some data here for a particular stiffness, we can, in our model, bend our kind of tail to different curvatures, figure out the energy, and back out the flexural rigidity to be that on kind of the same order of magnitude as the experiment, so that our elastic tail or flagellum has the right bending properties of a real flagellum. Okay, so we have kind of a force model, and then this is an extensibility, talked a bit about these stiffness parameters. Now I'm going to talk a little bit about this kind of bending portion. So what we're going to do is think about having our swimmer have a preferred curvature or preferred way to bend. What we mean by that is that, in a lot of experiments, they can track the beat form of these swimmers. It's, sinusoidal or potentially helical quasi-planar, but we know kind of that it beats with some type of propagating wave. And we've done, very basic models just to get an understanding of how everything's coupled and to really start to probe different model parameters. And we started with kind of a sinusoidal beat form where I have a constant amplitude B, is what I call kind of an activated motility. We've done some models with a constant amplitude or a tapered along the length. Or vice versa. But here we have kind of our constant B, we get a symmetric beat form. If we think about an asymmetric amplitude, we can choose that based on the local sign of the curvature so that it has a larger bend amplitude in one direction. And this is kind of our motivating experimental figures and our kind of weakly asymmetric emergent beat forms. And then we can also do something where we couple it to calcium. So calcium is known to cause these asymmetric beat forms and allow for hyperactivated motility. And here's a bull sperm, kind of two beat forms and some model simulations. And what we do there is we actually, instead of just forcing an asymmetry, we have a spatiotemporal calcium concentration, which feeds into this preferred amplitude. Okay. And this is one of the movies that I was trying to upgrade, but it didn't want to. So we do not have the movie. But in terms of the calcium, it's based on kind of a calcium signaling pathway. There's calcium entering through a channel on the sperm flagellum. Inside the flagellum, there's a few other reactions. There's calcium-induced calcium release from the redundant nuclear envelope. It's like an endoplasmic reticulum. But at the end of the day, what we do is we can track this calcium concentration, and instead of looking like a standard diffusion equation or reaction diffusion equation, there's a little bit of extra kind of intricacies, mainly due to the fact that this is a moving interface and we want to ensure mass conservation and other things. So we can track calcium, and then this, time-dependent calcium is what's going to feed into, it's going to feed into the amplitude. Okay, so here we have three different simulations. So this is three different simulations, similar to this previous slide or 2 slides ago. I can't get there. The blue, I believe, is constant amplitude. The black is kind of this asymmetric, a weakly asymmetric, and then the red swimmer is a different simulation where we actually account for that full calcium coupling. And what happens is when you have this asymmetric beat amplitude, which I don't know if I fully pointed it out on the video on one of the first slides, in, just a viscous fluid like water, a hyperactivated high calcium sperm is actually going to kind of do these circling patterns. So what we did for this kind of first initial model was try and parameterize everything. So we had the correct kind of emergence velocities or swimming speeds, curvy bilinear velocity, and made sure that we had all of the kind of swimmer properties accurately captured as best as we could in a very minimal model. And what we wanted to do after that is start thinking about, in this minimal model, can we start to tease out, what are key ingredients to allow for, self-organization or, hydrodynamic interactions or cooperativity of different swimmers. And there's been a lot of great, experimental studies over the years, as well as some really great modeling work. And so here I'm just highlighting two kind of different motivating studies of, sea urchin sperm self-organizing into these dynamic vortices, and then mouse sperm self-organizing into these long trains, where, a mouse sperm is say 100 microns and these sperm trains are, a millimeter in length. So there's hundreds of sperm there. Okay, so what we wanted to do is, understand if I have two filaments, do we get things like attraction, and can we also get synchronization of these swimmers? So here I have the initial starting point, kind of in these black dashed lines. And what we do is we actually start them out of phase. And then we continue to give each one of them the same preferred kind of beat form that's preferring them to be out of phase. And what happens is, due to the other swimmer, kind of each one's feeling the other one, what they do is they, hydrodynamically, they feel each other through the force field and the flow field. As we're kind of marching forward with our regularized Stokes-Lit simulations, they're feeling each other. Each swimmer is told to beat separately, but they feel each other. They kind of push apart due to hydrodynamic interactions. Then what they end up doing is starting to attract again. And you can see that even though I'm constantly telling them to have a different preferred curvature beat form, they eventually synchronize and attract. Similar to experiments by Woolley et al. And what we were trying to see here is, what are kind of minimal ingredients to allow for synchronization and attraction. And even if we tell these two swimmers to swim apart, kind of the emergent behavior of the model is that in order to kind of work with those force models and that energy configuration, it's actually more hydrodynamically efficient for the swimmers to beat in a synchronized beat form. And this just gives a sense of, for two swimmers, this is some experimental data where this is one sperm on the left-hand side, a second sperm on the right-hand side before they've synchronized or attracted. And then the middle is the synchronized swimmers, and those have a slightly higher swimming speed. And what we see here for our swimming speed is that I think everything's scaled by, so 1 is the swimming speed of a single swimmer. So when we have two swimmers swimming together, we can see that it kind of increases a little bit and then levels off, but it levels off at a speed greater than 1. So these swimmers start to attract, they get a little bit faster, and then they kind of synchronize and continue to swim at a speed faster than a single swimmer. Similarly, for two swimmers, we can see that, hydrodynamic efficiency of 1 is the single swimmer case, and we can see that the two swimmers that have kind of attracted and synchronized have an increased hydrodynamic efficiency. The interesting thing here is for kind of this third curve at the bottom. So we have these different bending moduli that we have in the model, these stiffness parameters. And for a lower bending parameter, stiffness coefficient, the swimmers are not able to propagate a beat form as nicely. They're kind of, right, the tails are less stiff, and due to that, they're not interacting as nicely in the fluid, so they don't see that increase in swimming speed and efficiency. Okay. So another thing we wanted to understand, so this was in collaboration with Julie Simons and Ricardo and Lisa, we wanted to think about for these hyperactivated swimmers, or the role of this increased calcium concentration and increased beat asymmetry. What is the role of this? So biologically, it's known that in order for a sperm to reach and fertilize the egg, in order for the sperm to reach and fertilize the egg, the sperm has to reach this kind of hyperactivated motility pattern. And there was a lot of question about, what is its role? Where is it playing a role? How do these particular swimmers or sperm, how are they able to reach and fertilize the egg, whereas others that don't achieve hyperactivated motility cannot? So one of the proposed mechanisms was that it could potentially help allow the sperm to continue to navigate when it's near walls, in particular the oviductal epithelium. So here we have, again, these are 3 different simulations. So we have kind of our high calcium, solving the calcium equations and having kind of that emergent beat form and amplitude based on the calcium concentration. Kind of this weakly asymmetric and this constant amplitude. And what you can see is that the high calcium case, if we kind of start them, if they're going towards a wall, if they kind of start close to a wall, they all kind of rotate a little bit. And the calcium one, due to that kind of higher calcium concentration and asymmetric amplitude, is allowed to kind of keep on making forward progress, where the other 2 swimmers kind of get stuck due to the satisfying that 0 velocity at the wall. But what's happening is, in addition to getting close to the wall, these sperm are interacting with cilia, they're near the wall, and there's really bonds that are being formed. So these swimmers, potentially, there's bonds being formed, and there's, this bond needs to be broken with a certain amount of force to kind of pull it away. So in a similar setup, we have these 3 swimmers. Again, the high calcium one is the red one, and the others are the slightly asymmetric or no calcium symmetric beat form. And we can see, different types of beat forms are being changed, different swimming patterns. And what's happening, and I don't have a plot of it, is there's kind of, as it gets close, there's these bonds or kind of elastic springs being formed. And the calcium one is the only one with that asymmetric beat form that's able to kind of break that bond and pull away from the wall. So if there's no bonds, it can kind of move along the wall. If there's bonds that could be broken, this increased asymmetry in that beat form allows the swimmer to pull away from the wall. Okay. So another thing, so this was kind of our basic setup, but another thing that we thought a bit about was, how do things change when you have these different fluid environments? And, these elastic structures are affected by, properties of the surrounding fluid. Beautiful work by Smith et al. For, how does the wavelength or the velocity of these swimmer change kind of in these different lower high viscosity mediums or these gels. Sperm and vaginal fluid. This is always a beautiful picture with kind of the bull sperm head behind here. There's this dense network of proteins that the sperm is actually trying to navigate through. So, a standard Stokes equation fluid, certainly being homogeneous, is not really capturing everything that's going on. So we wanted to think about, you know, in a really simplified modeling framework, how can we think about capturing some of these different types of fluids. And right, so there's you know, viscoelastic fluids. There's all different ways to capture you know, different fluid properties. We took the approach of using a Brinkman model. And a Brinkman model, there's kind of a few caveats. The main thing with the Brinkman equation is it's an effective medium approach. And even though I show this beautiful picture here, this is a very kind of dense network, but this equation is really representing a very sparse network of proteins. And instead of actually modeling each one of these proteins and how a swimmer is kind of interacting with each one of these proteins, we're taking that kind of homogenized approach and saying, you know, if you have some either randomly oriented or some kind of known network that's fairly sparse on average in this effective medium approach for these kind of sparse stationary objects, we can think about the extra resistance from those extra proteins or whatever they are in the fluid as being represented by this single kind of resistance term or damping term. Where we can think about this parameter alpha as being related to the inverse of the permeability of the fluid. Okay, and there's a few different ways to derive this equation. Another way we can do it is from thinking about a two-phase fluid with a stationary solid volume fraction. So a number of years ago, you know, there's this very nicely derived equation involving kind of these modified Bessel functions of the second kind that are relating the permeability of the fluid with volume fraction of fibers, fiber radii, and then we can kind of think about the corresponding alpha resistance parameter that would be in this Brinkman equation. And we can think about kind of what the volume fraction is, maybe collagen gel, cervical mucus. But again, I just want to say this volume fraction here, I don't know if you can see, it's like, 1-2%. So it's we're thinking kind of really in a dilute regime. And we can think about kind of the average spacing between these fibers and, depending on the different fiber radii and volume fraction, some of these fibers could be anywhere from say 20 to 100 away. And if your sperm or your swimmer is say 50 or 100 microns, there would be room for it to kind of swim through this array. And I guess the thing I didn't point out, and the reason why I like or find that this kind of Brinkman model is a great tool to kind of probe or investigate some of these effects of this fiber network, is that we have, what looks like Stokes' equation with just this extra term. So in the limit as alpha goes to 0, we're effectively back to Stokes' equations, whereas the permeability is infinite. Okay, and it's also a linear equation, so we can use all of our similar tools. So instead of using regularized Stokeslets, we can use regularized Brinkmanlets, basically just fundamental solutions to the Brinkman equation. Things get a little bit more complicated, but it's the same idea as method of regularized Stokeslets. So here is, alpha equals 0, we're thinking about kind of that Stokes regime, plotting the magnitude of the flow field. You can see these kind of large red and yellow regions, vortices going down the swimmer. For alpha equals 10, you can see they're kind of dampened out due to that kind of drag or resistance. And here you can see this is Stokes. And then as we're increasing either kind of the volume fraction or, fluid permeability, decreasing fluid permeability, we're getting an increase in alpha. And again, similar to when we had two swimmers, here this is a bunch of different simulations on one plot. Where each swimmer has the same exact kind of preferred beat form. And due to the extra kind of resistance in the fluid, this is the emergent beat form that occurs. What's interesting is that, there was some asymptotic analysis done, for kind of a 2D infinite sheet to say that the swimming velocity should basically be an increasing function of alpha. But what we observe, because we're in finite length, and basically this amplitude B as we go up in this alpha resistance parameter, we don't really achieve a larger amplitude, we achieve a smaller amplitude. So we actually see a smaller swimming speed here. And there's some kind of caveats due to the amount of work in the system and when or how you can achieve this. But, you know, for finite length, you have a small increase in swimming speed, and then it starts decreasing again, which is sometimes a little bit counterintuitive. So here things are kind of normalized. A single swimmer is, in Stokes is kind of a 1. So anything above 1 is an enhancement in swimming speed. And depending on the bending stiffness of the swimmer, you can see an enhanced swimming speed for kind of a small range or a slightly larger range, and then it decreases. And again, the main reason that we don't see the asymptotic results is because our amplitude quite often is decreasing. You're not able to achieve the larger amplitude. Okay. You know, and I guess the interesting thing here, as I said, is that you do get this enhancement for a small region. So it's kind of counterintuitive, because one would think that if you have these, you know, fibers or extra proteins in the fluid, likely they would be slowing down the swimmer. But there is this kind of sweet spot where the resistance actually works with the swimmer to allow it to get this enhancement in swimming speed. The other interesting thing is that in this model, we can think about, you know, this asymmetric swimmer. We can think about the asymmetric swimmer as being kind of this high calcium case. And this is a figure I showed earlier. This is highly asymmetric beat form with calcium and something like water. And then this is the same highly asymmetric, or was highly asymmetric, but high calcium. And in a gel or a polyacrylamide gel, we see that there's a real dampening of the beat form with kind of some small kind of amplitude bending right at the end of the tail. And here we have this same model. Again, each swimmer is given the same exact model term in terms of wanting to beat with a certain asymmetric amplitude. All we're doing is switching kind of this resistance parameter, and you get these different emergent beat forms and swimming speeds, but you do see in kind of the higher resistance or more proteins, which could be, you know, a very simple crude model of a gel. You see kind of the very dampened beat form with this kind of small oscillation of the tail at the end. Okay, so everything I showed you here, these were actually just kind of dots put on here for illustrative purposes to know kind of which way the swimmer was going and what we thought of as the head versus the tail, but really the head was just like the point on the flagellum. We can do models, you know, it's just more computationally expensive to actually model, you know, a cell body or head on these swimmers. And we've done that and started to kind of look at pairs of swimmers, and there's some, you know, really interesting dynamics and how things change from kind of Stokes equation to kind of these Brinkman models where you have potentially this volume fraction of these fibers characterized in this alpha parameter. So for two swimmers at low alpha or kind of in Stokes, you can, depending on the amplitude of the beat form, so this is kind of lower amplitude, higher amplitude, you can get two swimmers. For lower alphas or for Stokes, you'll get two swimmers that'll attract, as I showed before, with or without this cell body. The dynamics of how they tract become a little bit different. As you increase alpha, it's harder for the swimmers to attract. With no cell bodies, they kind of just stay a little bit apart. When they have the cell bodies, they do try and come together, but then they kind of push apart and go their own way. So we can look at kind of and characterize how the heads attract. In general, in most of our model simulations, head always seems to come together first, and it takes a little bit longer for the tail. And in general, we do see kind of some enhanced swimming speeds, but of course, based on the parameters. I guess I should say in all of these are kind of steady flow when we start them. If there are perturbations, in the fluid flow, we would imagine that most of these attractions would not be as stable. Okay. And then we can, start to understand if you were a swimmer with a cell body, what would happen for 1 versus 2 swimmers, and then what would happen if you're near a wall. So here, this is in 2D. I don't have a method of images. I just have an elastic wall that's kind of a wall of immersed boundary points or springs that are fairly stiff. And when you have no wall, in these models, the swimmer is going to kind of go pretty straight. When you start having a wall, depending on where it's starting at, it will start to attract. But as you have kind of an increased number of proteins or volume fraction, you lose that tendency to attract to the wall. And then over here, you can also see when you have 2 swimmers, they would just kind of attract with no wall. But once you start having the wall, kind of in Stokes' regime, you'd have the top swimmer attracting down, the other one kind of going to the wall and kind of coming up with some new kind of configuration, but you don't have as much wall attraction when you have the extra proteins in the fluid. Okay, so for that portion, what I wanted to say is that, right, we have ways with these kind of simplified modeling approaches to start understanding some of these emergent swimming speeds, kind of the emergent flagellar waveforms, when we might see interactions such as alignment or attraction or synchronization, and whether or not it's hydrodynamically efficient. Another thing I wanted to tell you a little bit about today is some parameter estimation work we've been doing. And, mainly thinking about, in a lot of these models, they're very simplified models. So you can think about these parameters, a lot of them as kind of a physical interpretation, a numerical interpretation, different ways to approach it. And then for this type of modeling framework, what is the correct parameter for this given model? On the other side, we have, these kind of interesting beat forms. We have these interesting beat forms, and we observe attraction or synchronization under certain conditions. And we might want to understand, under which parameters do we observe these type of phenomena? So instead of kind of running a number of simulations, trying to really back out what are the right parameters. So we started thinking about, can we, given some noisy set of data, can we actually understand what the parameters are in these kind of coupled fluid structure interactions where we have these very elastic and dynamic microswimmers? So you can get different types of data. You could get data for the exact kind of beat form, but we started even kind of cruder and thinking just about kind of CASA data, where computer-assisted sperm analysis, where we're just getting kind of a cell body that's being tracked at 20 to 60 hertz. Things are going in and out of focus, so quite often it's noisy. And we really wanted to think about, in a simplified modeling framework, can we actually get back some of these parameters? So we started with kind of this Bayesian setup where we have data, but we assume we have data that comes from some model, say G, and it's a function of certain parameters, theta. And with this model and our model predictions, we should have some error and then get back to our data. And if we use Bayes' formula, we can think about kind of this posterior distribution that's mapping output data back to our inputs theta and should be proportional to kind of our mapping of our inputs, as well as the likelihood of the observed data, as well as the prior distribution on parameters. But what's good to know is that for this prior distribution, we do have a sense of, for particular parameters, we do have from biological data and kind of interpreting what our model is actually capturing. We do have most of the time some type of range where we think these parameters fit into, and we can leverage that to be our priors. Okay, to do this kind of Bayesian setup and to keep things nice, we have to assume our prediction error is normally distributed, zero mean and covariance. I'm not sure if this assumption is totally valid in a lot of the data that one might get from CASA, but we assume that. And once we do, we can think about kind of the likelihood of our observed data. And the likelihood is, looking at this kind of nasty equation here, but this J term is a weighted measure of fit between model predictions and measured data. So we have our data, and then we have our model predictions. We are going to have to do this for a fairly large number of model predictions. So if we keep on solving our model for kind of our forward model to get these predictions, we can plug it into this J to then get our likelihood and start calculating these posteriors. Okay, and again, to kind of get a sense of, for these models we have, or the data we were assuming we had was just a noisy trajectory of CASA data, say from the location of the cell body. And assuming that we have the location of the cell body, we have this noisy data. It's a time series of data. And what we're doing in this modeling framework is we're running our model simulation, getting out kind of what the time series is from our model with a particular set of parameters, and then calculating out kind of posteriors and likelihoods for those given parameters. So we have the entire time series. We're running it through our model a large number of times, but kind of with fancier Metropolis algorithm and kind of optimal sampling techniques. We can sample a large range of values to parameter values to kind of map out the likelihood of these different parameters. And when we do that, if I start with some noisy head data and I assume I have some kind of uniform prior on, say, resistance parameter and a stiffness parameter Kc, what we can do is figure out kind of likelihood of different parameters. And here the data comes from alpha equals 1. So you kind of see these yellow spots in here around alpha equals 1. And I believe Kc was 8. This stiffness parameter. So what we get is kind of a visual mapping for a particular noisy data set, giving us the likelihood of certain parameter combinations. So what we can do from 20 hertz or 30 hertz, very sparse and noisy data, we can determine what particular parameter combinations could potentially give that data. We were really interested in trying to understand if I had some swimmers that could, attract and synchronize, if I had really crude data of just kind of head and tail trajectories, would we be able to also back out different parameters? Here, these kind of likelihoods or parameter values are a little bit less clean. But you still see, because of the noise and the two parameters, it's not as clear. But what you do see is these distinct yellow hotspots, which correspond to the actual parameter values that the noisy data came from, so we can recover those. So, what we've been able to do is start thinking about 4 different fluid structure interaction models. Can we use these Bayesian approaches with noisy data to recover parameters in our model? And the answer is we're starting to get there, and we think this is potentially a good approach to do that. Okay. and these movies aren't working. But in my last minute or two, I just wanted to say, so that was kind of a little bit about some parameter estimation we're doing, but myself working with the former postdoc Ming Hao, we've thought a lot about different ways to come up with faster methods for large numbers of structures and points. And we did some work with kind of kernel independent fast multiple method for method of regularized Stokes slits. And we can now kind of simulate a large number of structures in this manner. And Ming Hao cleverly developed some interesting preconditioners when we're doing kind of the inverse, where we know velocity and we're solving for forces on these structures. And we've also been thinking a lot about kind of, instead of really 2D or 3D filaments, really thinking about kind of these Kirchhoff rod representations of these swimmers. Okay, and I know I'm out of time, so I'll just kind of fast forward to the end if I can. Say we've been doing some things with calcium and all other fun things, but in the interest of time, I will stop there.